genome structure in an organism can be like a library of cassette. We can say genomes that make up from different chromosomes are similar to videotape. So if we have a library of videotape that may comprise the genome and each videotape correspond to chromosome 1, of course the tape itself is also correspond to the DNA. And when we play the videotape, what will happen? The expressing DNA and genes, so it will lead to synthesis and production of enzyme, protein, and so forth and so on. So I think now we are in the mitochondrial and chloroplast genome. Last time we talked about this and we asked actually question about this. The circular genome are the notable exception to the rule that eukaryotic chromosomes are linear. So however, this nicely fit into the phenomenon that eukaryotic chromosomes are linear and bacterial chromosomes are circular because those organisms seem to have evolved from the entrapped bacteria. Very, very important part. Please, I don't know what you want to do, but memorize here the human genome is composed of gene, 30% gene or gene related sequence, and extragenic 70%, so 70 to 30 is 100%. So please remember the human genome is divided to gene 30% and extragenic 70%. The gene itself is a coding region 3%. Intron, promoter, pseudogenes and gene fragment is 27%. So extragenic is repetitive is 15%, unique is 55%. Repetitive itself is a transmally repeated or intrasparse repetitive. Transmally is a mini satellite, okay? Mini satellite is a telomeric or hypervariable. And satellite, we have also, here we have micro satellite, okay? So okay, we go to the RNA. Okay, I want to say about RNA something that we didn't discuss last time. You should know that in the flow of genetic information, RNA is transcribed from DNA and from DNA and RNA also translated to protein. So in the flow of genetic information. For now, I will tell you about the RNA. RNA is a single-stranded molecule and would have a uracil as a nitrogenous base instead of thymine. When RNA transcribed from DNA, there can be different types of RNA. Okay, you see here, there is a single-stranded, but this one is a DNA is a double-stranded. Okay, so single-stranded, it has a uracil instead of thymine here. It's a ribonucleic acid, it's a deoxy. Okay. When it's transcribed, they have a different type, such as mRNA, as it carries information translated into protein. tRNA, this is the adapter between mRNA and amino acid. rRNA is a part of ribosome and participates in the protein synthesis. Small RNA is a used splicing and other cellular processes. For mRNA, this is how typical mRNA looks like. Okay. What you can tell us about this? We didn't say you last time. There's some student asking me. So this is how typical mRNA look like. So what are you seeing here? So if it's the typical structure of the gene, this one, DNA, and this is the typical structure of RNA. In the typical structure of the gene, what I am seeing? I am seeing the UTR. What is UTR? Anyone, what is UTR? Is untranslated region. I am also seeing promoter. By the way, that's what I want to say. how this is started? Many books say that. Do you know there is a RNA polymerase? The RNA polymerase will go inside from the area of the promoter. Okay, will go inside and it will divide this two stranded of the DNA, and it will make it will be start from 5 prime, never forget, starting from 5 prime. I don't know, I will show you this afternoon the picture that the RNA polymerase, if it's the RNA polymerase, is really entered to the double stranded DNA and they separated it like a zipper. If it's a zipper, if you think that the DNA is a zipper, something that separates the two zipper. So when it's separated, it's a time that RNA will fit itself with the following base which is available in the DNA. So for example, if a DNA base is a, the RNA base will be what? That's important. That if it's A, will be not T anymore, will be U. Okay, that's only different. So in this picture is already still is in the nucleus as far as I know, the mRNA. So what is important here, you are seeing a 5 prime cap. If somebody asks you in examination in the board exam of your country, that what is the 5 prime cap compound of or composed of? Okay, Siki, I ask from Jerin. Jerin, what is the 5 prime cap? What is inside that? Dennis, what is inside the 5 prime cap?
Phosphate, no?
No, inside the 5 prime cap is guanine. Okay? It's a guanine, it's a base. How about, I cannot see this. Okay, how about in the tri prime, Dennis? What we have in the tri prime? We have lots of adenine. Okay? That's why it's known as poly A tail. Please never forget. This is very important. They may ask it. Okay. So let me move forward to tRNA. tRNA is about 60 to 95 nucleotide long, include specific nucleotide. The hydrouridine, pseudouridine, is a clover leaf-like secondary structure consisting of a stem and three. Look, so why I am calling it anticodon? What is the really job of tRNA? The base adapter. The larger loop include a specific nucleotide triplet, bind to complementary codon in mRNA, display tertiary L-like. Actually, it's a T-like, but we are calling it L-like. So, what is the, do you think important here about anticodon? For example, if let me give you a good example here. You see that AUU, ACC. Okay, for example, here is the ACC in my mRNA. Okay, so my mRNA pairs here, for example, it's like this. Okay, there's the ACC. What is meaning of tRNA as an adapter molecule? So tRNA coming, tRNA is something like this. Okay, it's an anti-codon. So if codon is A, what will be in the tRNA? What will be? Can you somebody tell me? If it's A, what will be the tRNA codon? What is the base pair of A?
Thymine, doc.
Very good. Thymine. You should, all of you should know already. What's the base pair of C?
G.
Very good. G, okay. So this is a meaning of anticodon of tRNA. Okay, you should really already learn that because do you know what? I don't know about your experiences, right? In different schools you've been. But I told you one thing in the beginning of the biochemistry class. I think Jaren knows. Jaren can really be testify or justify what I am saying. You are coming from different schools to our school, Kulias College of Medicine. What is important here is you can always see that in all over the country, not country, all over the world, nobody will not sit down here and repeat you 100 times. Nobody. But I am telling you many, many times because I am expecting you become master of this. It's not only about this topic. I am going to teach you whatever my faculty teaching you. If you don't understand it, I will also teach to you 100 times. But I have expectation from you. You learn it. If I teach 100 times and 100 times you just be online and you just you just turn off my voice and not listen. So what is the purpose? Do you get my point? My Indian student, my goal is to pass their respective board exam. Our Filipino students must pass the PLE. They should pass. There is no excuse for them to not pass. And other nationalities, we have an American student here. She's always present. That's why I really appreciate this person. She should pass USMLE. We should dream big because our CEO has a big dreams also for us, Dr. David Pillay. So we should also dream big. That's why I am always saying this. We should make our administration to be happy, to be proud, because they give us whatever we want. So please bear with me. Please try to understand it, what I am teaching. So do you see this one, what I was saying, the L like? Actually, it's like an inverted L, diba? It's actually T like, but book, I don't know why books calling it L like. L like tertiary structure. But I am remembering it as this look like a T. That's why we are calling it tRNA adapter molecule. And if you see it here in the tri prime end, there is a where amino acid attached, right? The 3 prime end is a where amino acid attached. By the way, what is the first amino acid that my body will produce? If you recall, that is what? Let me see if we have it here. No. So the first, we don't have here. Okay. The first amino acid that my body will produce is coming from combination of A, U, G. It will give us methionine, right? Methionine, and remember the stop codons. The stop codons is UAA. I remember it as a URAV, UAG. You are going, and UGA, you go away. So these 3 codon, they are a stop codon. So when we reach here, we will stop. So stop. So because the RNA starts from 5 prime, it will stop in 3 prime because they reach to this one of this stop codon. So transcription will stop there. This is the stop codons. Okay, that's important. So you should remember only this one, AUG, because the first one, which gives us methionine, is the first amino acid, and you should remember this 3. as a start codon. So tRNA is stem ends in the tri-prime by the sequence of CCA attachment site for an amino acid. Each tRNA is a couple to the amino acid in the accordance with its anticodon. See, I was telling you about anticodon. But the coupling between the given tRNA and the corresponding amino acid is catalyzed by a specific amino acid tRNA synthetase. A specific amino acid tRNA synthetase. Okay. rRNA is a major constituent of ribosome. Ribosomes hold the mRNA in place, matches the anticodon of the tRNA carrying appropriate amino acid to the complementary codon of the mRNA, catalyzes formation of peptide bond, consists of two subunits of different size containing rRNA arranged with a specific protein. Both rRNAs and associated protein are slightly different in the prokaryotic versus eukaryotics. You are seeing the pictures here. Remember the sites, A sites, P sites, E sites. P sites the peptide or polypeptide. A is the amino, the first one which will be there, and E sites the exit. So microRNA is actually the new, and this is what we are showing. And you see the microRNA. MicroRNA binds to the messenger RNA and it's make the protein assembly to be block. It's a job of microRNA. It's make protein assembly to be block. It's a vital part of genetic regulation with enzyme origin, target of 60% of all genes, abundantly present in all human cells and are able to repress hundreds of targets. Conservation in organism ranging from unicellular algae to mitochondria. Small interfering RNA is a class of double-stranded RNA molecule, 20-25 nucleotide in length, play variety of roles involving the RNA interface pathway where it interfaces with the expression of specific gene, also acts as the RNA-related pathway, such as antiviral mechanism or in shaping chromatin structure. Okay, so this end of our lecture. Please attend Dr. Francesca lecture, and if you able to learn from her much, great. And please do not take one of the bad attitude of students. Do not make a drama. Doctor, we have class later, we are tired, if you don't want, don't attend. Okay, do not make drama to the faculty because faculty has nothing to do. She is just there to teach you. If you want to listen, listen to her. Some student was telling her, we are tired, we are like, do not do that. After her lecture, we will have review. If her lecture finished 3, we will have 3. And it's not mandatory to you attend. Even 10 students attend, I will continue. That other 2 topics is last time we are reviewing. Okay, so if she ends 3:30, we will start 3:30. Depends whenever she ends. But normally she is ending in 90 minutes. Okay, just when she ends, I will make the Dennis to inform me that Dr. end already, so we will start immediately 15 minutes after she ends. Okay, thank you very much and have a good lunchtime. Thank you, bye-bye.